okay, or else also called proton, H plus. Okay, in any solution, it donates its H plus ion or the proton. Okay, and bases are the substances which accepts the hydrogen ion, or else any compound, any substances that also dissociates a hydroxyl ion, and that can further bind with H plus ion. That are the bases. Strong acids, strong bases, we already know what is pH. pH is defined as a negative logarithm of hydrogen ion concentration. Okay, and it has the inverse proportionality relationship with the hydrogen ion concentration. Okay, increased hydrogen ion concentration means decreased pH. Okay, and decreased hydrogen ion concentration means increased pH. Okay, these are the normal ranges of the different ABG parameter, arterial blood gas analysis, which I will be discussing in more detail in the upcoming lectures. For right now, these are just the values you need to keep in your mind. Okay, hydrogen ion concentration, pH, partial pressure of carbon dioxide, and bicarbonate concentration. Normal blood pH, okay, it ranges from 7.35 to 7.45. Okay, if it is venous blood, it will be more towards the 7.35 side. If it is arterial blood, then it will be more towards the 7.45. So keeping the range narrow between 7.35 to 7.45. Okay, it is the responsibility of the different buffer system present in our body. Okay, its responsibility is to maintain the pH level within this narrow range. Okay, even the small fluctuation in the pH range may have the adverse effect in our cellular level, also clinically. Okay, there are different adverse effects we see if any changes occurs in the narrow pH range as well. This is Henderson's-Hasselbalch equation. You need not know this in more detail. Just this equation, from this equation we can derive a formula, and that formula can be implemented for calculating the pH of any buffer system. Also, we can calculate the pKa value of any buffer system. Also, in order to prepare buffer, what amount of salt or base, okay, what amount of base or acid we will be taking for making the buffer of our desired pH. So these are the implementations, or these are the benefits, okay, usefulness of the formula Hasselbalch-Henderson's equation. Okay. So different acids we studied: volatile acid, fixed acids. Fixed acids are inorganic acids and the organic acid. Inorganic acids are sulfuric acid, phosphoric acids, and organic acids are lactic acid, pyruvic acid, etc. Volatile, we have only one volatile acid in our body, which is carbonic acid, and which is produced from carbon dioxide and water in presence of carbonic anhydrous. We have discussed this. How buffer acts? Buffer, it combines with the hydrogen ion and neutralizes the hydrogen ion, you can say. Okay, it traps the hydrogen ion, it accepts the hydrogen ion, one way. Okay, other way, if the hydrogen ion concentration is less or more alkali is being increased in cell or in blood, at that time it can also dissociate its hydrogen ion in order to decrease down the pH. If the condition is tending to be acidic, at that time buffer, it accepts the hydrogen ion, neutralizes the hydrogen ion. And if our blood pH or else cellular pH, if it is tending to increase, okay, at that time the hydrogen ion concentration is less, okay, because less hydrogen ion means increased pH means alkalosis. In that condition, the buffer system, it will again dissociate, or it will donate its hydrogen ion concentration in order to drop down the pH back to normal. Okay, this is how the buffer works. So composition of buffer, we have seen the types of buffer. These are generally chemical, termed as chemical buffer. Okay, generally buffer, blood buffers, they are categorized as chemical buffer and the protein buffer. Okay, blood buffers. So what are the blood buffers? The chemical buffers, we have seen carbonic acid bicarbonate buffer, which is most predominant extracellular buffer. Okay, again the blood buffer is protein buffer system, and in protein we have different plasma proteins which will be acting as buffer, and hemoglobin also act as buffer in RBCs, and also phosphate buffer which is a intracellular buffer. Okay, so we call this as a chemical buffer or blood buffers. Okay, and these are chemical or the blood buffers are the first line defense mechanism. Okay, if any alteration is about to happen in our blood, okay, these are the buffer systems which will respond, which will resist the change in pH by acting in a first line. Okay, and if their reserve is depleted, okay, if their concentration gradually drops down, and even then the pH is not corrected, then only our other systems, other buffer systems will be playing their actions, like respiratory buffer system and renal buffer system. Okay, so chemical or the blood buffers are the first line defense against the change in pH. We have all seen this classification of different buffers, different chemical buffers or the blood buffers. We have also seen the action, how they act whenever acid or the base component is added to bicarbonate buffer system, okay, phosphate buffer system, how it acts. Okay, we also discussed the protein buffer system, and the main focus in the protein buffer system was the presence of negatively charged or the positively charged group. Whenever the protein or amino acid is at neutral pH, or let's say in our physiological pH of the blood, okay, it exists as Zwitter ion, negative and the positive ion. This amino group will be existing as NH plus, sorry, NH3 plus, it will be existing as NH3 plus, and this carboxyl group will be existing as COO minus. Okay, so it has Zwitter ion, and depending on the surrounding pH where these proteins are present, it can act as acid or it can act as base. We have also seen this here, you can see. Let us consider this amino acid which is existing in our physiological normal pH, and when it is shifted to the acidic surrounding, then what happens is from the acidic surrounding, the COO minus will now receive the hydrogen ion. Okay, because acidic surrounding means there will be more hydrogen ion in the surrounding. So it will accept the hydrogen ion, and the charge gained by the amino acid will be the positive charge. So in the acidic medium, always the amino acids are positively charged. Okay, you have to keep this in mind. Okay, this might be one of the objective questions. Okay, in the basic medium, what happens? In the basic medium means they have low concentration of the hydrogen ion. So that's why the hydrogen ion is being donated from NH3 plus. You can see NH3. Okay. Also, COO minus will donate his hydrogen ion, and it will be negatively charged. So amino acid in the basic medium will be negatively charged. In the acidic medium, it will be positively charged, and it acts as a buffer. Okay, this due to the presence of this positive and the negative charge in the amino acid, it has the ability to act as buffer, especially in the proteins, in the plasma proteins, and most predominant plasma protein is albumin. So albumin will be showing its buffer action in the blood. Okay, so hemoglobin as a buffer, where does it act? Where its action site? Its action site is in RBC, and what it is helping? It is not all the amino acids which is present in the hemoglobin, especially the amino acid histidine, which has imidazole group, and among that imidazole group also a special nitrogen N2. It has capacity to donate hydrogen or else to accept the hydrogen. So histidine will be contributing in the action, buffering action of a hemoglobin in the RBC. We have all discussed this, how it is acting as a buffer in the RBC. Okay, we have also discussed the chloride shift, and we also discussed all of these questions. And in the previous lecture, I just introduced the respiratory regulation. Now, whenever the blood buffer or the chemical buffer system fails to maintain the pH, okay, at that time, the secondary action will be taken by the respiratory regulation. It is the second line of defense against to resist the change in pH. Okay, there are different respiratory centers present throughout our body, also present in the large arteries, but mainly the main one is the central nervous in the point or the respiratory centers which is present in the CNS, mainly in the medulla oblongata, which responds to change in pH. Okay, whenever our blood flows to that region, to the region of the medulla oblongata, there is a sensor, you can say. Okay, the pH sensors, those are the respiratory centers in the medulla oblongata that will respond to the change in pH, and therefore they can act as a pH will change. They can either increase the rate of respiration or else decrease the rate of respiration, which will be more clear in the upcoming slides. But the mechanism of action, how they will regulate the pH is either our rate of respiration will increase or else decrease, okay, depending on whether it is alkalosis or else acidosis. Okay, and if respiratory regulation its significance is also like whenever we have any disturbances in the metabolic portion, say the kidney, there is something problem in the kidney, and if kidney is not able to handle or balance the pH system, at that time also respiratory regulation is very important, which will be clear later on. Okay, here you see how respiratory mechanism, respiratory buffer system, we also call this as respiratory buffers. Okay, first were the blood buffers or the chemical buffers. In that, the chemical buffers were mainly carbonate phosphate buffer and protein buffers. And respiratory buffer system means the respiratory system, which helps in maintaining the acid base. Let us start from the tissue level. Okay, in the tissue level, as a result of the metabolism, there is production of CO2. Yes, it combines with the water which is already present inside the cell, most abundant element, most abundant compound, you can say. Okay, in the cell, water, it combines with water. You should always remember the enzyme here. Okay, carbonic anhydrase is the enzyme which will catalyze the reaction for formation of the carbonic acid. Okay, carbonic anhydrase is the enzyme. Soon after the formation of carbonic acid, it dissociates here. This carbonic acid dissociates into bicarbonate and H+. Here you can see H+ and bicarbonate. Carbonic acid will dissociate into H+ and bicarbonate. This H+, this H+ now binds with the oxyhemoglobin. Okay, so the role of hemoglobin is to carry oxygen. It has carried oxygen to the tissue inside the cell. It is bound with the oxygen. Now, as the hydrogen ion dissociates from the carbonic acid, this oxygen will go and bind to the hemoglobin by substituting oxygen. Okay, it will replace the oxygen. Once H+ will go and bind with the hemoglobin, oxygen is being unloaded, you can see. Okay, so this oxygen is utilized by the tissue again. Now H+ is bound to hemoglobin, so it makes HB. This H is for hydrogen ion and this is hemoglobin. Okay, and we also have the bicarbonate. Now bicarbonate, it diffuses out from the cell. It goes into the blood circulation and this is carried to the lung. Okay, again once the RBC reaches the lung, okay, again once the RBC reaches the lung, again this bicarbonate shifts inside the RBC actually. Okay, RBC and then into the lung, you can say. So inside the cell, inside the RBC, which is present near the lung. Okay, this, even though here you can see the representation of the lung, but actually the reaction is occurring in the RBC. Okay, this is just trying to show, this schematic diagram is trying to show you that this reaction is occurring near the lung. Okay, actually it's happening in the RBCs which are present in the lung at the region of the exchange area. Okay, the gaseous exchange area, these reactions are occurring inside the RBC. I have already shown you when I was discussing this role of the hemoglobin, how it acts as a buffer. This is the same reaction, just the picture has changed. Okay, so this reaction is being displayed in the lung, but actually it's occurring in the RBC. Okay, you see. Bicarbonate will again diffuse inside the RBC. Now hemoglobin is also inside the RBC with the hydrogen ion. Now what happens again when the oxygen it start moving inside the RBC? What happens is this oxygen will now displace the H ion, H proton. Okay, hydrogen ion will be displaced by oxygen, and again hemoglobin will carry the oxygen. It is a loading of oxygen. Here you see the unloading of oxygen, HbO2. Oxygen is being unloaded here. Here at the alveolar level of the alveoli, the oxygen is being loaded in the hemoglobin, and there is displacement of H plus. Sorry. So here you can see this bicarbonate now binds with H plus and again makes carbonic acid. This again dissociates into H2 and CO2, and finally CO2 is being expired out. This is how the respiratory system is acting or as a buffer, or it is how it's maintaining the pH of a blood. Okay, so it's mainly handling the carbon dioxide. Carbon dioxide is then component of a volatile acid. Yes, carbonic acid. Because carbon dioxide, when it combines with water, it makes H2CO2, H2CO3. Okay, so this is how acid is being expelled out, which is produced inside the cell is being expelled out via lung. So if there is any problem in our respiratory system, what happens now? What you can predict? So for example, you can say if you have hyperventilating, as a result of hyperventilation, what happens is you will expel out more amount of carbon dioxide. So if there is more expiration of the carbon dioxide, the carbonic acid level will deplete, and that results in alkalosis. So in contrast to that, if our lung is hypoventilating, it's less ventilating, at that time more amount of CO2 is retained in our body only. Okay? So more amount of CO2 is retained means it will produce increased concentration of the carbonic acid, and that will dissociate and make large concentration of H plus and leading to acidosis. Okay. So this is how our respiratory system is helping in expiring out the acid component. Mainly the volatile acid is being expelled out with the help of the respiratory mechanism. If there are any causes that will inhibit the lungs or hyperactivate the lung, at that time we see respiratory alkalosis or respiratory acidosis. That we'll also see in more detail what are the causes and how actually it's occurring. Okay. So what actually happens whenever the respiratory system is altered? Okay. There are various causes for the depressed respiratory system. It might be injury, it might be because of the sedative drugs, okay, or paralysis of the respiratory muscles. Okay, at that time what happens? Our respiratory centers are depressed. Okay, and as a result we have decreased rate of respiration, or we also called it as ventilation. Decreased rate of ventilation or respiration is the same thing. If we are less ventilating, what happens? Means we are not expelling out more carbon dioxide. So the partial pressure of carbon dioxide will increase in the blood. And you know, this carbon dioxide will make carbonic acid. Okay, when carbonic acid is formed, this dissociates into H plus and HCO3 minus, and this concentration will gradually increase, and that will lead to respiratory acidosis. Okay. Next, if there is activated respiratory system, whenever the respiratory centers are activated, like you see in case of high altitude or any febrile condition, okay, those are the causes. We'll discuss that in more detail, the causes of all metabolic and the respiratory type of acid-based disturbances. We'll see more in more detail later on. But then simply to understand how the respiratory mechanism acts, Okay, just to understand that, I've drawn this schematic representation. Okay, activated respiratory centers. The causes are several. Higher altitude also leads to activation of the respiratory system, or hyperventilation, you can say. So it increases the rate of respiration. So as the rate of the respiration increases, what happens is decreases the partial pressure of carbon dioxide. When partial pressure of carbon dioxide is decreased, it is directly connected to the production of the bicarbonate, therefore carbonic acid. So carbonic acid production also decreases, and carbonic acid less production means less production of H plus. Okay. Here I have missed the arrow diagram facing towards the decrease in the decrease order. Okay, so here I will put the decreasing order of hydrogen ion concentration, and that will lead to respiratory alkalosis. So in compensation, the exactly opposite will be occurring whenever our body compensates either respiratory acidosis or respiratory alkalosis. What happens actually? The reverse, in a reverse manner, the compensation will occur in the reverse manner. Like you say, if there is respiratory alkalosis, after respiratory alkalosis, our body tries to compensate it by the respiratory mechanism also. Also, there are buffer systems as a first line defense, and parallelly the renal mechanism will also be acting in order to correct the pH. Okay, but then after a time, the respiratory system which was disturbed initially, the respiratory system which was disturbed will itself try to correct the pH, try to correct the pH in like exactly opposite manner. Whenever we have the respiratory alkalosis, after a time of respiratory alkalosis, what happens now? The respiratory center will depress. Okay, because as a result of respiratory alkalosis, the respiratory centers will depress, because respiratory centers are activated whenever we have high hydrogen ion concentration. Okay, respiratory centers, when they are activated, is in presence of high concentration of H plus. And here you can see, in case of alkalosis, we have low concentration of H plus. So the respiratory center will depress, then the rate of ventilation will decrease, partial pressure of carbon dioxide will increase in the blood, it will increase the carbonic acid concentration, and finally it will increase the H plus concentration. And as a result, I'm not telling you that respiratory alkalosis will end up in respiratory acidosis. It's not that concept. But the respiratory alkalosis as a compensatory mechanism, this is the way how it compensates, and it will gradually increase the pH. Okay, sorry, decrease the pH. Respiratory alkalosis is already increased in pH. So this is the way how our respiratory mechanism will try to drop the pH back to normal. Okay, respiratory alkalosis, if it occurs, okay, how it occurs? It is this mechanism. Okay, it will be followed by this mechanism simply. Okay, this is the pathology, how respiratory alkalosis is occurring. Okay, after respiratory alkalosis, the body will try to compensate. There are various mechanisms. We have already discussed the first one. The first line's defense is our bicarbonate system. Okay, phosphate systems are there, and proteins are also there. And after that, like third line of defense, that is the renal mechanism will also act, because there is something problem. If there is something problem in the respiration, at that time the renal mechanism will be more active. But gradually, or after some time, this respiratory alkalosis, how it is corrected? It is corrected by this means, by this way. Okay. As a result of respiratory alkalosis, our respiratory center will depress, decrease in ventilation, increase in partial pressure of carbon dioxide, increase in bicarbonate carbonic acid concentration, and increase in pH. Increase in hydrogen ion concentration, and dropping the pH back to normal. Understood? Yes?
Yes, sir.
Okay. So the story for respiratory acidosis will be, this is the pathology. This is the biochemical basis of the respiratory acidosis, this particular, up to here. This leads to respiratory acidosis. Now, how it is compensated? Now, this pathway will be followed whenever compensation is to be done. After respiratory acidosis, what happens? For the compensatory mechanism, our respiratory centers will be activated, because I've already told you how they are activated. Respiratory centers, they respond to high amount of H ion, and respiratory acidosis means increase in H ion concentration, hydrogen ion. Okay, so that is why after a time, after a person, whenever after person suffers from the respiratory acidosis, what happens? Gradually, the respiratory centers will also be activated, the CNS respiratory system, respiratory centers, and as a result of increase in the activated respiratory centers, it will increase the rate of respiration. Means now more amount of CO2 will be expired out because of increased rate of respiration. More amount of CO2 will be expelled out. Now there is drop in partial pressure of carbon dioxide. It also decreases the carbonic acid concentration. Later on, the H plus concentration will drop. Okay, even though I have not marked here as a drop sign, but you take it as a drop in carbon hydrogen ion concentration. And finally, the respiratory acidosis will be gradually compensated by drop in the hydrogen ion concentration. Okay. So this is the same thing, whatever I have discussed here, it is the same thing shown in the diagrammatic way. Okay. So let us start from here. Homeostasis disturbed, okay, because of hypoventilation. Okay. There are many causes. We'll discuss the cause later on. Okay, what leads to hypoventilation? There are several causes. Maybe in the upcoming lecture, 2 more lecture classes, we'll be discussing the causes of all this. So for time being, you just understand, by any means a patient is hypoventilating, okay, and homeostasis is disturbed. Start from here. Okay, a patient is hypoventilating. As a result of hypoventilation, I've already explained, the carbon dioxide partial pressure concentration of carbon dioxide will increase in blood, and that will lead to respiratory acidosis. How? You already know. Increase in partial pressure of carbon dioxide leads to more formation of the carbonic acid, which finally leads to respiratory acidosis. Okay. Now, respiratory acidosis has occurred. Okay. This is the condition. This is abnormality. Okay. Now it will start. Our body will start the compensation mechanism, buffering mechanism. So as I have already told you, what will the first line of defense? It is the buffer systems other than the carbonic acid bicarbonate buffer system. It's also the carbonic acid bicarbonate buffer. Okay, so here it's mistakenly written different buffer systems, mainly the chemical buffer systems. This will accept the hydrogen ion concentration, and it will try to establish the homeostasis again. And parallelly, the renal compensation is also there. It will try to excrete the hydrogen ion from the urine, and it also generates bicarbonate ion, or it reabsorbs the bicarbonate ion. The renal compensation will be there. That also helps to re-establish the pH, okay, or in order to it helps in homeostasis of the pH. And also gradually after a time, the respiratory system itself, which was disturbed previously, will will try to compensate now. Okay, how it compensates? As I have told you, in respiratory acidosis, once respiratory acidosis happens, after that hydrogen ion concentration is increased, yes, and that will activate the chemoreceptors, okay, the pH sensors in the central nervous system, in the medulla oblongata. Also the arterial chemoreceptors will be activated, and as a result, what happens? There is increased rate of respiration. Okay, this is the respiratory compensation. What is the respiratory compensation? The rate of respiration will increase as a result of activation of the respiratory system, and that will gradually drop the partial pressure of carbon dioxide. And all of these 3 mechanisms, okay, starting from this, okay, starting from the buffer system, blood buffer system will also try to compensate. And as you can see, for a time being, the respiratory system was disturbed. That is why the renal system was the mechanism that was occurring. And after that the respiratory system itself is also trying to compensate the pH by decreasing the partial pressure of carbon dioxide. Okay, this is the way of homeostasis in case of respiratory acidosis. In case of respiratory alkalosis, so first we start from the disturbance. Okay, so how homeostasis of pH, for the pH is disturbed, it is because of hyperventilation. Let us forget the causes of hyperventilation for right now. For time being, so hyperventilation means we are expiring out more amount of carbon dioxide. Okay, that is leading to decrease in carbonic acid and decrease in H plus. Therefore, as a result, we have respiratory acidosis. Now, respiratory acidosis, what happens? The buffer systems will be acting again. The buffer system will donate its hydrogen ion in the blood, because alkalosis means the hydrogen ion will be less. So we need more amount of hydrogen ion from our buffer system. So buffer system will be contributing the hydrogen ion in the blood. Next, we have the renal mechanism. Okay, renal mechanism. Now, what its role will be in case of alkalosis? Exactly the opposite role what we just saw in case of respiratory acidosis. It was secreting or excreting hydrogen ion and regenerating or reabsorbing bicarbonate. In this, the exact scenario we'll see as a renal compensation. The hydrogen ion will be generated, it will be reabsorbed, and bicarbonate we have to secrete out or excrete out for compensating respiratory alkalosis. And finally, whenever these systems have done their action, gradually the respiratory system will also be corrected. Now, what happens? Respiratory alkalosis leads to decrease in the or inhibits the chemoreceptors, which are located in the CNS and the arteries. And as an inactivation or inhibition of the CNS, the respiratory compensation will be decreased rate of respiration. So as we decrease the rate of respiration, what happens now? Again we will gradually increase the partial pressure of carbon dioxide, and the homeostasis is restored with all three mechanisms. Is it clear how respiratory system helps in compensating, or how it helps in homeostasis of pH? Yes?
Yes, sir.
Okay, fine. So now we move on to the renal mechanism. Okay, how renal mechanism helps in maintaining the blood pH. You might not be familiar with this because renal section has not been discussed yet. So this is the general feature, morphological feature of a nephron, you can say. Okay, so here we have the Bowman's capsule. Okay, and here we have the cells, nephronal cells are there, making the lumen. Okay, the epithelial cells are present in this lumen or the tubular structure. Okay, and we have the proximal convoluted tubule, which starts here, the most proximal to the most close to the Bowman's capsule is the proximal convoluted tubule. Okay, and next we have the loop of Henle. This is called loop of Henle, which is thin. Okay, it has the descending limb also, and this is actually the loop of Henle, and which also has the ascending limb. This is loop of Henle. And we also have the collecting duct, which is the distal convoluted tubule and the collecting duct ultimately. Okay, so this is the tubular arrangement of a nephron system. Side by side, these blood vessels are also running along the side of the tubule. And finally, what happens? There will be exchange occurring in the tubule. Some of the substances is moving inside inside the moving to inward the lumen, which will be finally excreted as a urine. For example, creatinine, uric acid, it will move to the lumen. Okay. Also, it is filtered from the beginning also. Okay. Some of the substances which needs to be excreted are filtered here. Okay. And some also goes into the lumen by the process of exchange that occurs throughout the lumen, throughout the tubular system. Okay. And this tubular system or the nephron is also involved in acid-base balance. For time being, you just understand the different sections of the different part of the nephrons. Okay. Proximal convoluted tubule, which is the most closest part to the Bowman's capsule. We have loop of Henle. Okay. And we have distal convoluted tubule, which is very far from the Bowman's capsule. That is why it is distal convoluted tubule. And ultimately, we have the collecting duct. Okay, so this is general morphology of the nephron. Okay, so how renal system helps in maintaining the pH is we'll be discussing, but for now we will say that the urine is actually acidic in nature. Okay, even though it depends on in our diet, the pH of urine, it depends in what diet we are taking. If we are taking protein in increased amount, then our urine will be more acidic. Okay, because of production of different acid. Okay, phosphoric acid, sulfuric acid will be more produced when we are taking the protein diet. And mostly the pH of urine is acidic, slightly acidic. It is around 6. And if we are not consuming any like protein-rich food or any food which has the pH, alkaline pH, that will also lead to increase in the pH of the urine. Okay, so the range of urine, pH range of urine, it is from 4.4 to 9.5. But at the most of the time in normal condition, whatever diet we are taking normally, at that condition, it's around 6. So from this only you can understand that kidney has the tendency to excrete acid. Also, it has tendency to excrete base. Okay, by this, looking out the pH of the urine only will understand, because acidic or the basic means presence or absence of hydrogen ion concentration in any solution. So you can see the range, yes, 4.4 to 9.5. So 4.4 means if the pH of urine is 4.4, it means that our kidney is excreting more amount of hydrogen. It might be because of acidosis which is occurring in the blood. Okay, that is why kidney is excreting more amount of hydrogen, and which is appearing in the urine, making urine more acidic. Okay, and if it is 9.5 or you can say 8.5, at that time more amount of bicarbonate is being excreted, or else less amount of hydrogen is being excreted because of alkaline condition in the body. If anyone is suffering from the alkalosis, for example, at that time we don't need bicarbonate, we don't need alkali reserve. Okay, more amount of alkali needs to be excreted in urine. Instead, we need more amount of hydrogen ion in case of alkalotic condition. So at that time, the pH of the urine will increase, making urine alkaline. So looking to the pH of the urine also, we can sort of make the clinical picture whether the person is suffering from acidosis or the alkalosis. Okay. So now how the nephron or how the renal system helps in regulating the pH of blood, pH of body. Okay. So these are the main points. Okay, how kidney helps in regulating the blood pH. It excretes hydrogen ion. Okay, these are the main topics we'll see in detail. Okay, in the schematic diagram, we'll see how it is excreting hydrogen. It helps in reabsorption of bicarbonate. Okay, also excretion of acid through the intercalated cell type A. Okay, in the collecting duct. this happens from the proximal convoluted tubule only. We will start excreting hydrogen ion. Okay, and some of the hydrogen ions are being also excreted, or it will be exchanging actually in the intercalated cell type A in the collecting duct, okay, which is present in the collecting duct, intercalated cell type A. Okay, and at that time there must be presence of some of the buffers which will tend to decrease the pH of the urine. Okay, if the pH of urine will increase, or sorry, if the pH of urine will decrease more aggressively, that will result in the damage of the urethra and the cell, whatever cellular components present in the urethra will be damaged because of highly acidic urine. So as our kidney will start excreting hydrogen ion, we also have certain acids like phosphoric acid which will bind to this free hydrogen ion, and that will try to balance the pH of urine. Although the pH of urine is acidic in most of the cases, but it will be not that concentrated which will damage our urethra, okay, or the urethral cells present in the urethra. So what are helping in neutralizing that increased amount of hydrogen in the urine? So that will be balanced by different mild acids like phosphoric acid, and which finally forms tritable acids, and also the ammonium ion, okay, NH4, that will also try to balance the hydrogen ion. And kidney not only excretes hydrogen, it also reabsorbs hydrogen, and that role is played by intercalated cell type B, which is also present in the collecting duct. So overall, the role of kidney is to excrete hydrogen, reabsorb bicarbonate, and there also occurs the exchange of hydrogen. This is the true, you will see later on in the diagram, the true excretion of this step. Okay, and also there occurs the reabsorption of the hydrogen. If our body is suffering from alkalosis, our nephron will also reabsorb hydrogen ion. Okay, let's start from excretion of the hydrogen. Actually, this hydrogen, it is not the hydrogen which is coming from different fixed acid. Okay, let us first understand different section. Okay, this represents the blood. This is the epithelial cell. Okay, cell present in the proximal convoluted tubule, and this is the tubular lumen. So what let me show you what is it. Okay, so this section is the lumen. This one is the lumen. A lumen where the kidney, where the urine starts forming. This is the lumen, and parallel to the lumen will be the blood vessels, blood capillaries. Here we have the blood capillaries. Okay, and lumen is a hollow structure which is surrounded by cells. Here you see, this is the cells. These are the cells. Okay, so exchange happens in between the lumen, and the blood vessels, and in intermediate in between we have the cells. Okay, the same figure has been highlighted here. Okay, here you see the plasma. This is the blood portion. Okay, next we have the cells. This is the proximal convoluted tubular cells, and then next we have the lumen. This will be the representation of the section. If we got the section of the cell, you see in this manner. Okay, we have supply of the blood. Parallelly, the blood vessels are also moving. Okay, we have the cell. Then these are the cells, and surrounding cells. All of the cells will surround to form a lumen. So here the excretion, excretion of hydrogen ion means not the exact excretion of hydrogen ion which are fixed acid, This is not actually the excretion of the fixed acid. Whatever acid which is being produced in the proximal convoluted tubule by this reaction is being actually excreted, and this is helping in reabsorption of the bicarbonate. This is not actual excretion of the fixed acid, but the acid which is produced inside the proximal convoluted tubule. This is the hydrogen ion secretion, not actually excretion but secretion. Okay, so fixed acids are phosphoric acid, sulfuric acid. Okay, that acids are produced in the cell by the result of the metabolism, but these are not actually excreted in this way. We are just discussing whatever acid which is being produced in the proximal convoluted tubule, how it is secreted. In proximal convoluted tubule, we have carbon dioxide, water, carbonic anhydrase will catalyze the reaction. Carbonic acid is formed. It will dissociate into hydrogen and bicarbonate. So hydrogen, we need to excrete hydrogen. Yes, hydrogen will be excreted in the tubular lumen in normal condition. And at the same time, bicarbonate will now go back to the blood circulation, and it will stay as an alkali reserve in the blood or the plasma. Okay, which is reabsorption of the bicarbonate, at the same time excretion of the hydrogen ion. And for the absorption of the bicarbonate, reabsorption of the bicarbonate, we also need help of sodium. So sodium, what happens? The filtered sodium, the sodium which was filtered in the lumen, it will be reabsorbed in the proximal convoluted tubule. Again, it will move to the plasma, and along with the reabsorption of the bicarbonate, sodium is also reabsorbed. Okay, so in this picture, there is excretion of the hydrogen ion. Side by side, you can see also the bicarbonate being reabsorbed. Here is the explanation of same thing: excretion of hydrogen ion, and at the same time, generation of bicarbonate. Excretion of hydrogen ion, generation of a new bicarbonate ion is being generated, and it is being reabsorbed. The second mechanism is reabsorption of bicarbonate. This is not the generation, but the reabsorption of bicarbonate. So some of the compounds like sodium bicarbonate, which are being filtered through the tubular lumen, this is the essential thing. We need this thing. Sodium bicarbonate, we need sodium bicarbonate, okay, as an alkali reserve. So this is actually the reabsorption of the bicarbonate. The sodium bicarbonate, it will dissociate, and again you see in the cell the reaction is occurring. H2O plus CO2 in the presence of carbonic acid, it is dissociating into H plus and HCO3. Now this H plus now comes to the tubular lumen. Okay, in the tubular lumen, it combines with HCO3, again forms H2, CO3, CO2 plus H2O, CO2 moves inside. Okay, so there is a way, it is a way to reabsorb the bicarbonate. Okay, it is preventing the loss of the bicarbonate, this mechanism. Okay, what happens? This H plus is coming out from the tubular cell, binding with the bicarbonate, making carbon dioxide. This carbon dioxide and water also, it moves inside the tubular cell. Again, it makes H2CO3, and again there is bicarbonate regenerated here in the tubular cell, and which is being reabsorbed. So it is preventing the loss of the bicarbonate. So this is actually the reabsorption of bicarbonate in the tubular cell. Okay. In the first diagram, we saw the excretion of the hydrogen. At that time, there is generation of the bicarbonate. At the same diagram, in the same diagram, we see the excretion of hydrogen, reabsorption of the bicarbonate. In the next diagram, we see how the tubular cell or this renal mechanism is helping to prevent the loss of bicarbonate by reabsorbing the bicarbonate. This is how it is being done. Actually, it's not the direct reabsorption. There are several mechanisms, several reactions occurring in between, and then only finally we can regenerate bicarbonate. But actually, this is the way of reabsorption of bicarbonate. Whatever which was about to be filtered and lost in the urine is prevented. Okay, it is being reabsorbed back. Okay, We'll discuss in the next class tomorrow in detail, and then from tomorrow only we'll move in